Hello, my name is Peter Schneider, and I'm a vascular surgeon at the University of California, San Francisco. I'm joined here by several world-class experts from around the world. Tatsuya from Japan, Yann from France, Joakim from Sweden. And we are really excited to have this opportunity to talk about something that has been very, very important to the growth of our field of vascular intervention and limb salvage, and that is drug delivery in the lower extremities. As we all know, there's a lot going on with drug delivery right now in terms of how it should be done, why it should be done, what the results are, is it efficacious, is it safe? And that's really what I'm hoping to dig into a little bit here today. So with regards to the efficacy of paclitaxel, specifically paclitaxel-coated balloons, I know we've all had our own experiences, but I'm wondering, maybe, Yann, you could take the first shot at this. You've done several studies of the use of paclitaxel in France, and you've also had a view to the worldwide literature and what's happening around the world. What's your impression about paclitaxel efficacy in terms of delivery by balloon, drug-coated balloon?
About paclitaxel is not just about balloon. It could be also about stent, drug-eluting stents. Definitely, you know, in a well-defined population. So, for example, RCT about paclitaxel-coated balloon focus mostly about claudicant and short lesion. And definitely, when you compare paclitaxel-coated balloon versus POBA, we have a superiority in terms of patency and freedom from TLR for mostly all the study. For DES, paclitaxel polymer-based eluting stents, we have also similar outcomes. And recently, Gunnar TP released some data for more complex and longer lesion. What happened next, in a more pragmatic study, we didn't see this effect of paclitaxel eluting technology for larger population. And we have the debate here about the class effect and about also the quality of the lesion. Does it matter if finally to use this DCB or this DS? We should perhaps to take care more about the quality of the lesion, the length, the calcium loading, and probably also not just about the lesion, but also about the profile of the patient, claudicant versus CLTI.
And then, Tatsuya, how about in Japan? I mean, it seems like the Japanese clinicians were early adopters of drug delivery. What's been your experience? Is this something widely used or only occasionally used?
Thank you very much, Peter. In Japan, drug eluting technology is very widely used. According to the Japanese National Registry data, approximately 80% of the treatment was were performed with something drug technology devices. Right?
80%?
Yes, DS or DCB. However, the penetration, adoption of the DES is relatively low, right? Gradually decreasing because many physicians want to finish the procedure with balloon-based technologies. I think it's a simply Japanese physician tendency, and also better stenting and also combination strategy, DES and DCB is not allowed. Therefore, many of the physicians try to avoid the stenting. Then mainly, DCB is mainly used approximately 65% or something, then 15% were treated with DCB. And rest of the 20% of something is a bare metal stent or something. Anyway, so adoption of the drug eluting technology is very high.
Very high, I see.
Yeah.
In Sweden, You have the SWEDEPAD 1 and 2 trials. And of course, Dr. Nordanstig, you were one of the PIs. This was a massive undertaking nationwide in Sweden. You got to be proud about that. How do you interpret the efficacy findings for drug delivery, either by balloon or stent in SWEDEPAD? How has that affected practice, would you say, in Sweden?
So largely speaking, I think that the SWEDEPAD trials sort of mirrors what was known in the literature in the smaller device-specific trials, but the patterns are there. I think it's similar. So the TBR rates during the first post-procedural year was lower with paclitaxel-coated devices in SWEDEPAD 1. That's quite consistent what we expected. And the difference is that we targeted hard endpoints. So we tried to prove, limb amputation. So we dimensioned the trial for a 3% reduction in major amputation linked to paclitaxel-coated devices, and we were unable to show that. So I think that our contribution is moving from proved efficacy in a very controlled research environment on a specific device to a broad implementation as a routine procedure. And then the benefits might have been a bit overstated in that context. So that have resulted in our country so far, after doing this, that the use has been more limited. And I also think we might have underestimated this risk for distal embolization. And I think that could be one player here in our trial that we did not prove limb amputation reduction. We might be under-recognizing the fact that we have some problem with distal embolization. SWEDEPAD is not designed to determine the effects of DCBs versus DES. The best thing we could do is to show, a strategy of primary stenting and primary stenting versus PTA and bailout stenting with paclitaxel or not. That is something we can do, and we are working on that subgroup analysis. But we cannot, in our data set, derive direct evidence for DES versus DCB because it's a strategy trial.
Paclitaxel efficacy, I think, is not so clear below the knee, but seems very clear on many studies to be quite efficacious above the knee. And yet, in the SWEDEPAD series, it showed some efficacy at 1 year in SWEDEPAD 1 in the CLTI group. Again, many FEMPOP, but also many BTK patients. But in the claudication patients, where I thought this is the place where it would likely show efficacy, it didn't. And I don't know what do you think is going on there? Is it that it really wasn't efficacious and all the other studies are wrong? Or was it it was because it was a mix of devices, maybe some were more effective than others? Or the other possibility is that with the use of paclitaxel in those patients, there may have been so many other variables that weren't controlled, or, it just wasn't part of the study. I mean, this was although it's an RCT, you're covering a huge population, a broad population. You're trying not to turn patients away from the trial. I'm assuming you're trying to enroll patients. And most of the trials in the US that are done for IDE for approval of an investigational device, they're very selective. That patients with certain conditions, maybe they don't qualify for the study. You have your inclusion, exclusion. So maybe it's all of the above. I don't know. Do you have an impression?
Yes, so I think that's a great question, and I agree with you. I was also puzzled a bit by that. I think it's could be a matter of, health system context first. So it's a quite conservative approach to claudication in Sweden. So we advocate, exercise and best medical treatment as the first thing. We try to follow the ESVS guidelines. So patients that entered our trial with claudication, I would say it's fair to say they had really severe claudication. So that's the first thing with the selection of patients. But then our main efficacy endpoint was quality of life, and that was neutral at 1 year. But we also measured TVR, and that would be, an important endpoint to prove the biologic effect in our trial, because some other endpoints like patency we cannot show for every patient because we don't have ultrasound on all of them.
Of course.
So, and that's a limitation. But I do think that the decision for a TVR in a patient with claudication, it is not only based on the fact that there is a restenosis. It's a patient desire. It's the doctor's preference. Might be less prone to offer a re-intervention in a patient with an early failure that's still just claudicating because of a sense that you might do harm if you do a repeat procedure. So I think TVR and TLR are less suitable endpoints in claudication due to that fact that other reasons that what happens with the restenosis plays into the decision. And that's not the same in CLTI because it's more of a hard indication for re-intervention where we have restenosis in and perhaps recurrence of CLTI symptoms.